In the second segment, we will continue our discussion on the olfactory system with Dr. Kelleher. There seems to be a fascination with pheromones. Could you explain?
The pheromones are interesting because in reality, depending on whether you're a scientist or in the perfume industry, they mean two completely different things. By definition, the term pheromone was originally derived from insect studies, where an insect is able to release a particular chemical cue that's detected by a second individual of that species, and it elicits a particular behavior or physiological response. How that translates into vertebrates and mammals in particular is a little bit more difficult, and most scientists now believe that the term doesn't, and the definition in itself, isn't really that helpful in understanding how social cues are being processed. There's plenty of odors that are being processed as social cues that have different purposes and are probably not specifically pheromones. In terms of what people's interest in pheromones is, it's sort of been morphed by their interest in how social behaviors are elicited. And so the term has become known as being any type of chemical cue that is given off by another individual that has some sort of a behavioral connotation to it, or elicits some sort of a behavior. And specifically, people think of pheromones as being influencing reproductive behaviors or mate selection, or people will smell a pleasant odor around somebody and it's a pheromone because now I'm attracted to you because of your smell. And so in terms of understanding the science behind it, that term's not all that useful anymore. But in terms of understanding, or at least referring to things in social behavior, it's still used quite broadly. There's a lot of interest in pheromones in terms of certainly mate selection. We've done a little bit of research, and there's been quite a bit of research done on the topic of the major histocompatibility complex. It's called MHC for short. And what that does is it's part of our immune system. It's a specific part of our immune system in which the body is able to recognize self versus non-self. And so if you're to have an organ transplant, this part of the immune system can tell whether or not that organ belongs to you or whether it's a foreign object. And there was a theory years ago by Lewis Thomas who thought that, well, if the immune system can detect self versus non-self, is it possible that cues can be released outside the body that help other individuals detect self versus non-self? And there's been quite a bit of research now into the particular aspects of this major histocompatibility complex, or MHC, that allows animals, and quite possibly humans as well, to detect relatedness, whether or not an individual is closely related or not related at all, or some intermediate between. And it probably plays a huge role in the avoidance of inbreeding. And so why are you not attracted to somebody that you're related to? Well, in the case of animals, it's because of the odors that are produced by this system.
So pheromones tell us a lot about other humans and...
Absolutely. Odors in general. We have a large battery of odors that can tell us, and we could probably detect individuality, whether or not somebody is similar or different than yourself. Certainly we can detect whether or not an individual is similar or different from another individual.
So what's an example of an unconscious mechanism underlying pheromones?
So, well, in the case of rodents, for instance, we know that pheromones, or we know that these chemical cues appear to be processed in a somewhat different mechanism in terms of the pathways in which they're following in the brain and some of the areas of the brain that they actually project to. So some of these cues are not going to be projecting to the primary olfactory cortex, but instead taking a different route and eliciting responses directly in the amygdala, emotional centers, and also the hypothalamus, which is going to be an area that's going to be triggering all sorts of subcortical responses and behaviors. And so that would be the difference in terms of how those cues happen to be processed.
All right, and you already talked about this, but what main areas are thought right now to be involved with behavior involving pheromones?
So if people are detecting things that can be classified as pheromones, certainly it's going to be, again, the amygdala. It's going to be different areas of what we call the limbic system, as opposed to being detected and processed directly through the thalamus, as in a conscious relay. And there are certainly going to be hypothalamic pathways that are going to be receiving olfactory cues, which certainly could trigger those innate type behaviors in humans just as well as they do in other animals.
All right, I've also read about smell deficits. What is a smell deficit?
Smell deficit, we call smell deficit an anosmia. Anosmia is actually a complete loss of smell. There's hyposmias, which are decreased sense of smell. There's actually hyperosmias, which are increased sense of smell. The classic example is when women become pregnant. Oftentimes their sense of smell increases, and it's believed to be a hormonal regulation of it, but that's an increased sensitivity to different odors. Anosmia is, in essence, a complete loss of detection of odor cues, generally through that main olfactory epithelium, the normal olfactory pathway. Oftentimes, anosmics still can smell things in terms of, and even with some bit of odor quality through different systems, such as the trigeminal system, so pain sensation. So you wouldn't be able to smell a rose, but if someone were to take smelling salts and stick them under your nose, you certainly are going to be able to smell those. And so oftentimes people that lose their sense of smell, anosmics, will still have some sense of detection of chemical cues through their nasal cavities.
So what causes smell deficits?
So in humans, there's primarily three causes that'll elicit a loss of olfaction. The first would be a major infection in the nasal cavity. And in that case, you'd have a destruction of the olfactory epithelium. Oftentimes, that can actually be repaired. Olfactory neurons are unique in that they're one of the neurons in our brains that actually regenerate. We have basal cells that are in the olfactory epithelium that will form new cells. And just in normal occurrences, they turn over about every 60 days. So they'll have a complete loss of olfactory neurons sporadically, never at one time. You don't become anosmic every 60 days, just for a few brief days. But since those neurons turn over, if there's a destruction just of the neurons, you can recover within a couple months. Oftentimes smell will come back. If the infection goes deeper and actually gets those basal cells, those progenitor cells that are dividing, then oftentimes smell can be completely lost and permanently lost. Another occurrence that makes people anosmic oftentimes are nasal polyps, or some sort of nasal polyps would be not necessarily even growing right on the epithelium, but some sort of a blockage in the nasal cavity, which is going to redirect airflow. And oftentimes just removing those is a complete recovery. The other more common way in which people lose their sense of smell is actually head trauma. So when the olfactory neurons send their axons up to the brain, they do it through a bony plate called the cribriform plate. It's this porous bone. And so the neurons are located underneath, send their axons up to the olfactory bulb. So during head trauma, if the brain moves, and the olfactory bulb specifically moves inside, it'll sever those olfactory axons. Theoretically, you should be able to regain sense of smell if those neurons then die, new neurons are formed and send their axons back up. But what happens is oftentimes scar tissue or something will form on that cribriform plate, and those axons never find their way back up. And so if in the case of head trauma, that's one of the more severe times in which usually recovery is not going to be possible. But it's relatively a common occurrence in head trauma. I think about 10% of people that exhibit some sort of severe head trauma end up losing their sense of smell.
I notice whenever I'm sick, things smell a little different. Could you explain why this might happen?
It could be a number of reasons. Certainly the nasal cavity is full of mucus. The mucus forms a number of purposes, and in fact, one of the changes over from aquatic animals to terrestrial animals is now we have to take chemical cues and somehow take these airborne cues and get them back into a mucus or something that can be liquid and then bind to receptors from there. And so the mucus and whether or not it's viscous or thick or thin or what have you plays a huge role in how those chemical cues are brought into the mucus and bind the receptors. There's also going to be binding proteins in the mucus that are also important for presenting, but also taking away those chemical cues. Certainly changes in mucus is going to have a severe effect. It's actually thought that when a nasal cavity is wide open, the sense of smell is worse than when it's closed a little bit because of the redirection of airflow. If you have the nasal epithelium is only located on the very top of the nasal cavity. There's plenty of room and plenty of other areas in which air will flow through your nasal cavity without reaching that top. So having it a little bit constricted can sort of get that airflow up there and change the direction of how the chemical cues are going.
What could you say that we can learn from smell deficits about the olfactory system in general?
Deficits tell us a little bit about where in the brain these things can be processed. In the case of our research, we actually utilize smell deficits in order to learn things about behavior, because what we're doing is we're eliminating particular types of sensory signals or particular types of sensory organs. So there's, as we mentioned before, there's those 5 distinct systems, and then each of those also has subsystems. In the main olfactory system, for instance, in most animals, although it's a pseudogene in humans, there's a system called this GCD, or guanylyl cyclase D subsystem. And what that is it's an olfactory neuron that has a different signal transduction pathway than the other olfactory neurons. It's about 1 in every 100 neurons has this different signal transduction pathway. There's something different about the type of cue that it's sending. And we know very little about what that cue happens to be. There's some evidence now that it might be carbon dioxide that's being detected through that system, or carbon-related compounds, or possibly even gut peptides that can activate these guanylyl cyclases as well. And so what we do is we eliminate, genetically, we can eliminate different pieces of the olfactory system and learn exactly what those pieces are doing in terms of social behavior. Now, it doesn't always work well that way. You're going to have to introduce them again and integrate them, because all these systems are working together in the brain, in the nasal cavity, and then in the brain, in order to really understand how social behavior is going to be mediated. We have to study them all in whole, but we also have to study all the pieces individually as well, and then hope when you take those pieces and add them back up to the whole, you have something that is really in total the behavior you're interested in.
So a severe deficit in an animal, could it affect their success in their environment?
Absolutely. Well, if you take an evolutionary aspect or evolutionary look at it, in order for a species to be successful up to that point, they utilized all of that sensory system in order to get there. It wouldn't be around if it wasn't being utilized in some way or form. So for instance, the fact that the GCD is now a pseudogene in humans, whatever it happened to be that is detecting, maybe it is carbon dioxide specifically, because humans aren't very good at detecting low levels of carbon dioxide. Once it gets strong enough, we detect it as pain sensation in our trigeminal system, but at low levels, we can't detect low levels of carbon dioxide.
That's why we have carbon dioxide detectors. And,
Yep, carbon dioxide, carbon monoxide detectors to prevent you from asphyxiating on high levels of CO2 or carbon monoxide. And so certainly, in that case, other animals that still have the system are utilizing this system for whatever it happens to be. Behaviorally, we're not exactly sure. We have some good ideas is what behaviorally they're using it for, but we're not exactly sure. But for whatever it is, we obviously aren't using it anymore.
Well, thank you very much for coming in here. I've learned a lot about the olfactory system, and I think it's a very interesting topic, and I hope you enjoyed the interview.
Well, thank you very much for having me.
Thank you.